Hi friends, it's me Danashree again from Sri Science. And today, before starting our lesson, let's see an interesting fact about our Earth. Have you ever known that there is an eighth hidden continent? Yes, there is one. And that continent is Zealandia. Getting back, today we are going to learn the test for reducing sugars. Okay, what are these reducing sugars? Yeah, we have learned this before. We have learned that all the monosaccharides are reducing sugars, and disaccharides, maltose and lactose are reducing sugars, whereas sucrose is a non-reducing sugar. So let's see how the test is. Okay, first let's do this test for a monosaccharide and a reducing sugar, a hexose sugar, glucose. Okay, our aim in this test is to show that glucose is present in the sample solution. Okay, if we want to do a test, we need some equipments. Here also, we need some equipments. What are those? Okay, first one is a boiling tube. Why can't we use a test tube instead of this boiling tube? There is a reason. Because in this experiment, we're going to heat. So, test tube can't stand heat. Therefore, we are going to use a boiling tube. And our next thing needed is Benedict reagent. It was named Benedict'S reagent because it was invented by Benedict. And this is blue in color as it contains copper sulfate in it. Okay, and the next thing we need is the sample solution. This is the solution which we are going to use for the test, which means we are going to test whether glucose is present in this solution or not. And the next thing we need is a trough with water. And at last, we need a tripod with gauze on top. Now let's see the procedure. First, take the sample solution in a boiling tube of volume 2 cm cube or 2 mm or a volume as you wish. And next, you should add the Benedict's reagent into this sample solution. The sample solution is colorless, but the reagent is blue in color. So when you add the reagent into the sample solution, it turns blue in color. And note one thing, the volume of Benedict solution that you are going to add into the sample solution should be equal to the volume of sample solution you have taken. For example, if you have taken 2 cm cube of sample solution, you should add 2 cm cube of Benedict solution. Then mix it well. And next, this solution that you obtained by mixing the sample solution and Benedict's reagent should be immersed into a water bath, which means this boiling tube should be immersed into a trough with water. And then you have to place this trough in tripod and heat it using a Bunsen burner. Here we use the technique water bath because the heat transferred throughout the mixture of sample solution and Benedict's reagent should be equal. Then only we can get a clear observation. And next, here you should boil the solution or heat it up to 95 degrees Celsius. Why do we mention boil or heat it up to 95 degrees Celsius? Because you can observe the changes when this temperature rises up to 95 degrees Celsius. If you say boils, no need to say heat up to 95 degrees Celsius. But if you say heat, then you should mention 95 degrees Celsius. Because boiling means heating up to 100 degrees Celsius. so nearly 95 degrees Celsius. But heating means it can be like up to 40 degrees or 50 degrees or 70 degrees. Therefore, you should mention the temperature up to how much you should heat. Then you will be able to observe a color change. That is from the starting. Like our solution was in blue in color. Next, it will turn to green, then to yellow, then to orange, and to brick red. This is the color change. If you observe this color change, then you can conclude that glucose is present in the sample solution. Note: this test will answer for all monosaccharides because you know that all monosaccharides are reducing sugars. So if you do this test separately for galactose and fructose also, you will get the same observation. Moreover, this test will answer for disaccharides, maltose and lactose because they are also reducing sugars. But this test won't answer for sucrose because sucrose is a non-reducing sugar.